Hi, thank you for attending today's webinar. Today I'm going to walk you through the Fluidigm Advanta Dx SARS-CoV-2 RT-PCR assay. We hope that you find the webinar interesting today, and this application is for high-throughput COVID-19 diagnostic testing using saliva, and Fluidigm with this assay has received emergency use authorization from the FDA. My name is Mark Lynch and I'm going to be your presenter today. The agenda for today's session really falls into 5 different buckets. We're going to walk you through from start to end what this assay is all about. The assay, as a reminder, is the Advanta Dx SARS-CoV-2 RT-PCR assay. We're going to start off by talking to you about how the samples are collected for that assay and how that compares to other testing mechanisms. We're going to explain why saliva is actually an optimum testing input method for mass COVID-19 testing. We'll then go into some of the details about the Advanta Dx SARS-CoV-2 RT-PCR assay and how it works. And of course, we have a situation where we've got new strains appearing all of the time, but we want to take a moment to explain how these new strains are targeted and detected with the Advanta Dx SARS-CoV-2 RT-PCR assay. And of course, hopefully, once we've walked through all of those 4 items, you'll be asking, how do you get a test? So we'll talk to you about some of our programs that we have in place that will enable you to get a test. Let's start with the collection methods. We've got a collection method that uses saliva as an input. We want to talk to you about how that saliva compares to other so-called swab-based testing mechanisms. On this slide, we really talk about traditional sample types and the challenges that they have. At the start of the pandemic, the most common used collection method was a nasopharyngeal swab, or an NPS. This is shown on the far left of the slide. When collecting with an NPS, there are significant drawbacks to both the patient receiving the collection and the healthcare provider acquiring the collection. For example, the sample must be collected by a trained healthcare provider. There's significant PPE, personal protective equipment, required, and gloves must be used and changed for each patient. There's risk to this in the approach because what actually happens, as shown in the cartoon on the right-hand side, is that the swab actually goes right into the back, right into close to the base of the brain. So there's actually a risk that the patient would cough, sneeze, or vomit. And it's really very much a technique dependent. The person collecting the sample has to be highly trained and know exactly what they're doing. And this, at the start of the pandemic, was considered the gold standard for COVID-19 testing. As we start to go through the pandemic, and for the reasons that we've just described, anterior nares swab became more commonplace, ANS. ANS is less invasive to NPS. It is faster than NPS, and actually it can even be self-administered or self-collected. But it is still technique dependent, and it does have a lower viral load of detection sensitivity compared to nasopharyngeal swabs. The take-home message of this slide is that these may be traditional sample types, but they do have challenges. Fluidigm and the Advanta Dx SARS-CoV-2 RT-PCR assay uses easy to collect saliva, and that saliva performs comparably to most commonly used NPS tests. The cartoon, again shown on the left-hand side of the slide, shows that the saliva for the Advanta Dx SARS-CoV-2 RT-PCR assay can be collected in any sterile tube. This means that it's more scalable to mass population testing. It means that it's non-invasive because the patient can very easily deposit saliva in this non-coated and sterile collection tube. It means that you can improve the sample acceptance rates, and because our test does not call for an extraction of RNA from the virus, we do not require any collection agents like preservatives to inactivate the virus. Simply, the patient can deposit the saliva directly into the collection tube, and that collection tube can then go to the lab for testing. And as you'll see later on in the presentation, there's evidence in the publications, of which there are many, of equal or better performance compared to nasopharyngeal swabs. And in fact, a collaboration that Fluidigm performed with a real-life setup in New York showed that we had 100% agreement between saliva and nasopharyngeal swabs. And we'll show that later on too. The key message here is that saliva does in fact have numerous advantages over nasopharyngeal swabs. And these are highlighted on this slide. We bucket these into these categories of accuracy, convenience, and safety. Accuracy is shown in the teal color at the top and is the first bullet point, where saliva quality is unaffected by the collection process. In other words, all you have to do is deposit the saliva in a collection tube. And because of the way in which the Advanta Dx SARS-CoV-2 RT-PCR assay works, there's no need for a large volume of saliva, which means that if you are collecting saliva from elderly population or young kids, the volume doesn't really matter. If you compare that to nasopharyngeal specimens, if they're collected by an inexperienced personnel, the quality of that sample may not be adequate enough to yield a result. So from the point of view of accuracy, saliva is a method that will improve the accuracy of testing. The next bucket of advantages really relate to convenience, and these are shown in the purple typeface, or the second bunch of bullet points on the slide. The collection of saliva is convenient, stress-free, and non-invasive, and multiple specimens can be easily collected from the same individual at the optimum times required. Saliva collection is advantageous for individuals with physical or mental handicaps, and collecting saliva allows for measuring a range of other useful analytes at the same time in the same sample. So going forward, there would be a capacity to be able to look at other substances within the saliva sample. You'll see that the saliva is stable at room temperature for extended periods of time. In actual fact, the Advanta Dx SARS-CoV-2 RT-PCR assay is stable at room temperature for 5 days. And the saliva collection does not depend on any material that is in short supply. And in actual fact, the workflow with the Advanta Dx SARS-CoV-2 RT-PCR assay actually eliminates certain parts of the workflow that are in short supply, such as the extraction of RNA from the saliva. And the last piece in terms of these advantages for saliva versus nasopharyngeal swabs really relates to safety, which is shown in the pink color at the bottom group of bullet points. And that is a low risk of exposing labs to hazardous samples. Saliva samples can be obtained while social distancing, and saliva collection could reduce the need for healthcare provider personnel protective equipment. The take home here is that there are numerous advantages of using saliva compared to using swab-based testing. So let's look at this in a little bit more detail. What we've gave you so far is the benefit of saliva over using swab-based testing. What I would like us to do now is to just delve into a little bit about what the communities are saying about it, the scientific community in particular, as to why saliva is an optimum sample type for mass COVID-19 testing. So as part of our workflow, what we do is we look at how the publications are coming out in the community and compare them through the process. And we look at this, for example, with saliva performance in the community compared to swab-based testing. So what we're showing you here on this slide really is that on the far right, we're looking at a particular study which was published relatively recently in 2020 that is looking at saliva compared to nasopharyngeal swabs compared to anterior nasal. These saliva were collected from adult patients that presented at a drive-through test center with symptoms suggested of COVID-19. The samples, the subjects, were instructed to swab both nostrils, along with a pool saliva in their mouth, and then repeatedly spit a minimum of 1 ml of saliva into a sterile tube in the presence of a healthcare worker. And then the nasopharyngeal swab was actually collected at the last in the sample sequence. And we had 1,104 paired specimens collected from 368 unique patients. And what we're seeing here is that the number of positive specimens, when you take these three different types of sample collection, were highest in saliva. 81 in saliva versus 80 in nasopharyngeal swabs versus only 70 in arterial nasal. The data in this publication helps to back up some of the advantages that we outlined in the previous section of the presentation. The other dynamic to consider when we look at the literature to look at how saliva compares to nasopharyngeal and anterior nasal is really the sensitivity. And so in this slide, what we're doing is a summary of the comparison of all the studies that have been published, and we're comparing saliva to nasopharyngeal swabs. We're looking at sensitivity. For those interested, sensitivity is defined as the number of positives of one specimen type divided by the total number of positives by both types. In the absence of a true reference standard, all positives are therefore assumed to be true. So when we do this, and you can see this on the table at the far right of the slide, when we do this, when we compare saliva being more sensitive to nasopharyngeal swabs, we find that that happens in 52.6% of the studies performed. Saliva being equal to nasopharyngeal swabs happens in about 10.5% of the studies performed. And when we look for nasopharyngeal being more sensitive to saliva, that happens in 36.8% of the comparisons in the studies performed. The key take home on this slide is that from a sensitivity perspective, we find that in the peer-reviewed publications and literature of saliva being more sensitive to nasopharyngeal swabs or equal to nasopharyngeal swabs, that it is around the case in 63% of the times, or 63% of the studies. The key take home here is that in 63% of evaluated studies, there is demonstration that saliva is equal or higher sensitivity than nasopharyngeal swabs for the detection of SARS-CoV-2. So we've looked really at the kind of advantages of saliva. We've looked at how it compares from the sensitivity perspective. How does it compare now from the economic perspective? Again, the key take home here is that we looked at this in a meta-analysis of 37 independent studies. These 37 studies had 7,332 paired samples, and they were reviewed against a reference standard of a positive result on either sample. And the sensitivity of saliva was 3.4% points lower than that of nasopharyngeal swabs. But because this is an economic analysis, the study showed that collection of specimens by saliva compared to nasopharyngeal swabs would save over $600,000 per 100,000 persons sampled. The costs for nasopharyngeal swabs and saliva were calculated to include all the material costs: swabs, transport media, containers, personal protective equipment, and estimated labor cost. So if the prevalence of SARS-CoV-2 is 1% among sampling, NPS, nasopharyngeal swabs, would identify 79 more positives, but it would equate to $8,093 per additional identified infection. So the conclusion here again is that saliva sampling seems to be similarly sensitive to swab-based testing, but it's a good economic alternative to nasopharyngeal swabs for SARS-CoV-2 testing. So we've looked at cost, we've looked at sensitivity. What about detection rates? We did the same type of study with detection rates, comparing saliva and nasopharyngeal swabs from a detection rate perspective. And what we've found here is that when we look at the literature and the data sets within that literature, we find that saliva specimens have a higher detection rate than nasopharyngeal swabs in 71% of comparison. And nasopharyngeal specimens had a higher detection rate than nasal swabs in 82% of comparisons. Again, the key message here is that we're not saying one is better than the other, but what we're trying to say to you is that using saliva as a testing mechanism for COVID-19 is ideal for mass testing. So with that, we'll now move on to a little bit more about the assay itself. We've looked at an introduction, why saliva is a good testing mechanism. We now need to consider what the assay looks like itself from a lab perspective. What are you getting when you spit into that tube? And for that, I'd like to introduce to you the Advanta Dx SARS-CoV-2 RT-PCR assay. The Advanta Dx SARS-CoV-2 RT-PCR assay is an accurate, efficient, non-invasive saliva assay that is designed to meet the global need for a rapid diagnostic test for SARS-CoV-2 infection. Key points on this assay. Point number one, this is a saliva-based test. This means that the patient would deposit saliva into a non-coated collection device at a small volume and be able to then send that collection device or take that collection device to the lab for processing. As we're using saliva, we're also using an extraction-free methodology. What that means is that normally, under many other workflows, you would perform an RNA extraction from the viral material, basically taking the virus, isolating the virus, and collecting the nucleic material to then process and get the results from. Our test goes directly from the saliva. There's no need to perform that extraction step, which helps eliminate any problems with bottlenecks of supply chain, and it also helps to improve the workflow efficiency. The third important point is that we have lower sample volume requirements. Our workflow is a microfluidic based workflow, and that uses just 15 microliters of sample input. To give some perspective to that, the average person can produce between 400 microliters, which is close to a mil, and 1.4 mil of saliva. So with the Advanta Dx SARS-CoV-2 RT-PCR assay, we use just 15 microliters of that sample input, meaning that it's optimum no matter what the age group is. We also use, as part of our molecular chemistry, 10 times less than what is used in a micro-well plate, which is important again because it means from a cost perspective, we have advantages, and it means that from a supply chain perspective, we use less reagent, so it means that your reagents will last longer and you're unlikely to fall into any supply chain constraints. The last point is that we have a high test capacity. Our platform is modular, which means that it's scalable. And you can go from anything from a few hundred tests per day up to 6,000 tests per day, depending on lab setup and lab throughput. These are the key points to remember for the Advanta Dx SARS-CoV-2 RT-PCR assay. The cartoon at the bottom of the slide that I will briefly walk you through also explains a little bit about the workflow. You saw this cartoon shown in step one before earlier in the presentation. This cartoon is the depositing of the saliva in any non-coated sterile collection tube. That saliva can be deposited in this collection tube and then stored at ambient temperature for up to 5 days. That's the end of the patient involvement in that sample. This sample then makes its way to the laboratory, and the laboratory workflow kicks in from step 2 through to step 8. And to break this up, because it looks like a lot of steps, we'll split this up into pre-processing, which is steps 2 and 3, and microfluidic workflow, which is steps 4 through 8. So the saliva sample arrives at the lab and we have to pre-process that. That takes place in step 2 and 3. This involves heat treating the samples at 90 degrees for 10 minutes, and that then follows an extraction-free, directly from saliva, reverse transcription and pre-amplification. Reverse transcription, meaning that we take the saliva and the particles within that saliva and we directly convert that to the nucleic material that's detected in the microfluidic workflow. At the same time of that, in the same step, without needing to change any tubes or plates, we perform a pre-amplification. That pre-amplification takes place with 2, not just 1, but 2 COVID markers, N1 and N2. And the pre-amplification also takes place with a control marker, RNase P. We perform that small pre-amplification step because our microfluidic workflow requires the number of molecules to be high enough to be sensitively detected at a microfluidic scale. After steps 2 and 3 are complete, the pre-processing is complete and the samples make their way to the microfluidic workflow. And the microfluidic workflow takes place in steps 4 through to 8. Step 4 involves transferring the samples and the markers of interest into a microfluidic device. This microfluidic device is the same size as a traditional microwell plate, meaning that it is compatible with any lab pipetting instrumentation, whether that's manual pipettes, 8-channel pipettes, or liquid handling robots. The microfluidic device is then ready to be auto-loaded using one of our controllers, which are shown in step 6. These controllers do slightly different things, but in the case of the Advanta Dx SARS-CoV-2 RT-PCR assay, they do the same thing. And they're going to move the samples and the assays into the square in the middle. In other words, it's going to start to compile the nanoliter reaction volume reactions. And that takes place in a hands-off fashion. And once that's complete, it then goes into the Fluidigm real-time PCR platform, which is called the Biomark HD, where a fast real-time PCR reaction occurs. We have also our Fluidigm interpretive software, which is used to call the results. And depending on lab setup, every 35 minutes, you'll generate data from step 7 to step 8 for 192 samples. Here's our workflow in a little bit more detail for those that are interested in it. You can see on the left-hand side the hands-on time, and you can see on the right-hand side in teal the instrument time. And what this schematic is showing you is a little bit more detail of what I've just mentioned on the previous slide. You can see that the same schematic exists, but what we've done is gone into much more detail with regards to the pre-processing, which is shown here at the dilution step, the heat treatment step, the reagent preparation step, and the assay preparation step. So all of these 4 buckets that are shown here are the pre-processing steps that I mentioned. These take place in steps 2 and 3. From this point on, where the cursor is now, loading the 192.24 IFC, thermocycling and exporting the results, this step here is all the microfluidic workflow. This is taking place from steps 4 through to 8. And again, just to provide you with a little bit more detail on what's happening at each of these steps. As mentioned at the start of the presentation, the Advanta Dx SARS-CoV-2 RT-PCR assay has received emergency use authorization from the FDA. And to do that, Fluidigm performed both an analytical validation and a clinical validation. And what we're going to walk you through now is the analytical validation. The analytical validation involved taking 3 saliva pools and spiking in different concentrations of SARS-CoV-2 from the ATCC. What we found when we did this spike in and heat inactivation was that the Advanta Dx SARS-CoV-2 RT-PCR assay exhibits an LOD, or limit of detection, of 6.25 genome equivalents per microliter of sample. And what you can see is that we have in the table below, we have different concentrations of the ATCC viral particles from 50 target level genome equivalents per microliter of sample down to 0.391 target level genome equivalents per microliter of sample. And you can see that when we're measuring the N1 and the N2, because please remember the Advanta Dx SARS-CoV-2 RT-PCR assay measures both, when we see that, we start to see that our dropout occurs around about the 6.25 genome equivalents per microliter of sample. In other words, for N1, we get a 95% detection rate. For N2, we get 90% detection rate. And for the internal positive control, RNase-P, we get 100% detection rate. And this LOD is confirmed whether you use an RX controller or the Juno instrument to load the IFC. All that means is that no matter what the loading mechanism is for the microfluidic device, you get the same LOD. We also performed our clinical validation, and that clinical validation was performed in collaboration with Washington University School of Medicine in St. Louis. The initial clinical validation involved comparing results to nasopharyngeal swabs. And our data with Washington University School of Medicine, which was performed on 77 retrospective blinded saliva specimens, of which 43 were positive and 34 were negative, were collected at one clinical site and tested with the Advanta Dx SARS-CoV-2 RT-PCR assay. These specimens had matching nasopharyngeal test results with an FDA EUA SARS-CoV-2 real-time PCR test result with nasopharyngeal swabs. All of these 77 specimens were processed as per the instructions for use that are available on our website and included the interpretation of the results based with a crossing threshold CT value cutoff of 32. The key take homes here really is that we have 100% positive percent agreement and we have 100% negative agreement when we do this comparison between saliva extraction free using Advanta Dx SARS-CoV-2 RT-PCR assay and the EUA comparator using nasopharyngeal swabs. You can see that when positive in one, they're positive in the other. When negative in one, they're negative in the other. And so out of the 77 samples, we have 100% result agreement in saliva extraction free compared to nasopharyngeal. Now, because the Advanta Dx SARS-CoV-2 RT-PCR assay has been on market for some time, we actually received our authorization in August last year, we've also done other studies. We've done studies comparing saliva to anterior nares. And we actually did this as a Fluidigm-led multi-site concordance study, where outpatients presenting to a high patient volume clinic provided both an anterior nares swab that was tested using a high complexity RT qPCR assay system for SARS-CoV-2 at an independent testing facility. The saliva specimens or samples, were collected from the same individuals to test using the Advanta Dx SARS-CoV-2 RT-PCR assay at Fluidigm. And in this study, what we were actually able to find when we compare saliva with extraction free, shown in purple, to anterior nasal swabs tested with a comparator, shown in teal, we found that there were 5 positives that were identified in saliva that were not found in anterior nares. We also found that there were 6 negative samples that were found to be negative in the anterior nares, but positive in the saliva test. And so it meant that what we had in total was that from a positive percent agreement, we had 100%, but from a negative percent agreement, we actually had 98.7 in the total 460 sample set that we studied using this. So what we find is that 460 specimens tested on both platforms. We had 3 saliva specimens that arrived empty, and so we had a very low sample rejection rate at just 0.64%. 449 specimens with negative results were found on both platforms, but there were 5 anterior nares swabs and saliva samples that tested positive. So we have 100% positive percent agreement. There were 6 subjects, however, who tested positive only with the saliva specimens. And we're in the process of confirming the 6 discrepant calls using an orthogonal method. But one line of thought is that the reason that we're detecting these 6 positive samples with saliva is because we're able to pick up lower viral loads in saliva than what is possible in anterior nares. The Advanta Dx SARS-CoV-2 RT-PCR assay is an assay that is extraction-free and is available to be run in numerous labs across the United States. You can see here the product specifications. Maximum number of samples per run is 192, but the modular nature of our platform enables up to 6,000 tests to be run in a 24-hour period. The protocol time and the system time are shown here, and these are saliva specimens collected by a healthcare professional. It's a kit-free extraction procedure, and we use just 15 microliters, 1/5 of saliva. The limit of detection is 6.25 genome equivalents per microliter of saliva, and we test 3 targets, 2 for COVID, i.e. N1 and N2, and 1 for RNase P. The detection chemistry is FAM-based, and the internal control, as mentioned, is RNase P. We use thermocyclers to heat inactivate the virus up front, and that is the Applied Biosystems Verity platform. As mentioned, we did this as a collaboration with Washington University School of Medicine in St. Louis, and we co-developed the assay and performed the clinical validation with the teams at Washington University School of Medicine in St. Louis. Both our collaborators that we worked very closely with have very good things to say about the Advanta Dx SARS-CoV-2 RT-PCR assay. In other words, there's close collaboration between the teams at WashU and Fluidigm to help develop the kit, and the folks at Washington University School of Medicine state that such a test could help overcome supply chain bottlenecks that have limited testing for COVID-19 and help identify infections. Another angle is the kind of lab-based angle, and that lab-based angle really states that the Fluidigm test doesn't require RNA extraction, which is a time-consuming and expensive step necessary for other types of tests using SARS-CoV-2. And the team at WashU have stated that the test that we developed with them could be easily scaled up and made widely available. In the last parts of the presentation, we'd like to cover off on how we detect the new strains with the Advanta Dx SARS-CoV-2 RT-PCR assay, and of course, how can you get a test? Because what we've seen is that there's a lot of new strains that are coming out. There's the South African strain, the UK strain, the Brazilian strain. There's going to be more and more of these strains, and commonly we get asked about how the Advanta Dx SARS-CoV-2 RT-PCR assay can handle those. So let's just talk a little bit about the strains to start with. The viruses undergo strong immunologic pressures in humans, and it can accumulate mutations to outmaneuver the immune system. And so these mutations currently form all the time in viruses, but the ones of interest, the variants of concern, currently are the United Kingdom variant of concern, as well as the South Africa variant of concern. And these really seem to impact specific regions within the N area of the virus. And so what we've found is that with our Advanta Dx SARS-CoV-2 RT-PCR assay, we use the US CDC primer and probes that target 2 regions in the nucleocapsid protein. That's these 2 here. Okay, so this is the actual nucleotide that we target, and that our assay is unimpacted by these new strains. In other words, our assay can be used to make sure and detect the presence of the virus independent of the strain. So if we look, for example, at the nucleocapsid protein, the N1 assay, there's actually the mutant change here that takes place. It goes from GAT, shown in red, and then it goes to CTA. And what we're finding is that the first base position of the N1 forward primer begins at position 28287. And therefore, amplification of the first Fluidigm assay, the N1 assay, would not be impacted by this substitution. The same is true for the N2 assay. The N2, the nucleocapsid protein change that takes place, the base substitution for C28977T changes nucleotide position 28977. And this first position of the N2 forward primer begins at 29264. So again, amplification of the N2 assay would not be impacted by this substitution. The key take home here is that when you run the Advanta Dx SARS-CoV-2 RT-PCR assay, then you're able to still measure the virus independent of the strain. And as we have more and more strains coming online, Fluidigm continues to look in silico at the impacts of these new strains. And so far, with all the strains that exist out there today, the South African strain, the Brazilian strain, the UK strain, we have not found that any of them impact the performance of our assay. In actual fact, the N1 and the N2 assays that we use to detect COVID will detect COVID no matter what strain the subject presents with in their saliva. The last part of the presentation is really how can you get a test run today? What we've went through so far is a very technical presentation, but if you're listening to this and you say, okay, I want to get one of these tests, how can you get that? Well, Fluidigm has a network of labs, and what we do is really connect our customers to deliver results. So examples include, for example, employee testing or workplace testing or pop-up testing sites. Whatever the community wants, that saliva can be collected and then the testing process can begin. We've got a network of labs shown here in the center of the slide that is spread across the United States that can actually run these. And these labs are hospital labs, they're private labs, or they're commercial labs, or they're university labs, for example, and they can all be equipped or are equipped in many cases to run the tests. And then we have partnerships such as with Advanta, where the data analysis and the results can be sent to those providing the saliva samples. And if you actually look at the Fluidigm website, we have a Community Connect program on our Fluidigm website that enables you to be geared to where you need to be with regards to COVID testing, whether that's campuses, university parents, K through 12, skilled care facilities, return to work testing, or sports or entertainment. With our Community Connect program, which you can see here, we have labs already established that can run the Advanta Dx SARS-CoV-2 RT-PCR assay. In summary, what we presented to you today is the benefit of saliva as a mechanism of testing. We've also walked through the comparison of saliva to nasopharyngeal and anterior nares. We've walked through the technical parameters of the test, i.e. the Advanta Dx SARS-CoV-2 RT-PCR assay, and we've explained how that assay can still measure the virus independent of the variant that the subject has in their saliva sample. And finally, we've explained using our Community Connect program how you can actually get a test. With that, I would like to thank you for your time. It's been my pleasure to present to you today the Advanta Dx SARS-CoV-2 RT-PCR assay. With that, we'll pause for question and answer session. But as a reminder, if you need any further information, you can contact one of our demand generation executives in the details shown on this slide. Thank you for attending today's webinar, and we'll now pause for questions and answers. Thank you. So right now I'm just checking through to see what questions we have coming in on our chat box, and we have a few that have already came in through the webinar. So the first was really the question relates to, can you recover and sequence the amplified products in order to distinguish between the variants? And it's actually a very topical question because what we're seeing is a lot of our customer labs are indeed being tasked with sequencing the positive samples. And as such, the workflow for that currently involves generating a positive test using the Advanta Dx SARS-CoV-2 RT-PCR assay, and then going back to that sample and actually following our library preparation microfluidic workflow to be able to then sequence those positive samples. We haven't touched upon the library preparation workflow in this presentation today, but that workflow has the same type of value as the workflow that we described to you today. In other words, it uses microfluidics to be able to prepare libraries in batches of either 192 samples, 96 samples, or 48 samples, which is a lot higher throughput than some of the other library preparation protocols in 96 well plates, for example. Some of them are only 24 samples at a time. So in actual fact, our labs are starting to get that request to sequence for those strains, and the workflow in which they do that is as I've just outlined in this question. Another question that's came in is how many COVID markers does the Advanta Dx test assay for? And it's actually 2. On the presentation that we just delivered, the markers for COVID in the Advanta Dx SARS-CoV-2 RT-PCR assay are N1 and N2. So we actually have 2 markers in our test. Some of the other tests on the market actually only assay for 1, in particular some of the other saliva tests. But the Advanta Dx SARS-CoV-2 RT-PCR assay assays for 2 of the COVID markers. And if you look on our website at our instructions for use, you'll see the different combinations of the N1 and the N2, i.e. the COVID markers, and what constitutes a positive result. And you can see that in our IFU. Another question that's came in is related to the Community Connect program. Where exactly are these network of labs? So we've got labs spread across the United States for the Advanta Dx SARS-CoV-2 RT-PCR assay. And as mentioned, they're a combination of commercial labs, i.e. that you just send the samples to and they run them. And there's also a network of university and hospital labs that we utilize. If there's a specific area in the US that you want to reach out to us on, you can contact our colleague Jason Zog, who's one of our demand generation executives, and he'll be able to point you in the right direction as to where those labs are specifically located. Another question that's came in is related to, so the other question that's came in is related to the amount of saliva that's required. What if populations do not produce enough saliva? Can the test be used for this? So as mentioned in the presentation, the assay itself, the Advanta Dx SARS-CoV-2 RT-PCR assay, uses just 15 microliters of saliva. And what we've found is that that is a very small volume compared to what the average person can produce. The average person can produce roughly 1 ml of saliva. So the Advanta test that we have here at Fluidigm uses just 15 microliters. So it literally is like a little passive drill that's required inside the collection tube. And that 15 microliters can then go right into the lab workflow to use the assay and run the assay. So a very small volume, and it means that it's particularly applicable to care home communities, younger folks that don't produce a lot of saliva. And again, actually, in our instructions for use, we outline the guidance for that also. Another question that's came in is, what is the difference between your sensor chip and other microfluidic sensors done in silicon? Okay. Yeah, we never really went into that in much detail. The chip, the microfluidic device that we use for the Advanta Dx SARS-CoV-2 RT-PCR assay uses Fluidigm proprietary valve technology. which basically means that we've got valves that open and close, right? So you apply pressure to some of the lines in the microfluidic device and it compartmentalizes the reaction. Actually, the benefit of that is that you can parallel assay all at the one time. And in the particular case for COVID detection, that 15 microliters of saliva, we're actually assaying N1, N2, and RNase P all at the one time. So for the diagnostic kit, it means that that saliva sample is not split up into different reaction vessels. That's very important because it means that the saliva, small volume of saliva, actually goes a long way using the Fluidigm microfluidics with valves. The other important part of using the valve-based microfluidic technology of Fluidigm is that if there's other markers that someone wants to look at, now we're maybe talking less about the diagnostic part of this, but more about the kind of capacity to build an LDT, then for example, if you wanted to look at not just N1, N2, but maybe look at RSV, flu A, flu B, or actually look at specific mutations, then actually you can add them onto the microfluidic device. There's space to do that on that side. And the benefit of that and the benefit of the valves in being able to do that, again, is that you don't have to split that sample up. In other technologies, you'll have to split the sample up into different reaction vessels, or you'll have to perform multiplexing reaction chemistry, meaning that you would have to put the same marker in the same well and optimize for the most abundant marker. And the microfluidic devices of Fluidigm don't require that because we actually use the valves to parallel assay from a single sample at a time. The other benefit of that also is that it does not increase the cost. So you would think normally I'm going to add extra markers, it's going to increase the cost. It doesn't do so because of that parallel processing on the microfluidic device. Another question that's came in. Since it's such a small volume of saliva, could you just swab in the mouth and then centrifuge the swabs to collect liquid? It's actually a very good question. Obviously, that is separate to the instructions for use for the Advanta Dx SARS-CoV-2 RT-PCR assay. But the short answer, if I maybe use the kind of more research angle, is yes. Okay, so as long as you have enough material to go into the reaction. Okay, so we do have labs that have developed their own LDTs, lab developed tests, to be able to do such things. And in actual fact, Fluidigm has a product actually called the Advanta RT-PCR Assay, which is an RUO product that many of the labs use for swab-based tests, right, with extraction. So there in fact is a workflow that enables that. And so if the questioner wanted to use, take a swab in the mouth, centrifuge the swabs and collect the liquid, that would be part of the RUO product. And actually the benefit of that would be, again, everything that I've highlighted related to the valves in microfluidics and related to the microfluidic devices themselves. And one final point on that is, in actual fact, you have greater flexibility too, because you could look at some of our other microfluidic devices that can process 96 samples at a time or 48 samples at a time. The next question that's came in is, can you inactivate the virus in the whole sample before you open the tube so the laboratory workers would not possibly be exposed to the virus? Okay, understood. Our current IFU, our instructions for use for the Advanta Dx SARS-CoV-2 RT-PCR assay, calls that the saliva arrives in a collection tube. That saliva is then stable for 5 days at ambient temperature. And the benefit of that is really for testing regimes such as in employee testing, or such as when you're trying to do batch testing for schools and stuff like that, so that if there's only a few samples, the lab can actually hold those samples for a few days, up to 5 days, and then heat inactivate. So the heat inactivation step is actually performed as part of the lab workflow and not part of the patient workflow. In other tests, there is a viral transport media inside of that inactivates the virus, but the challenge with that is that you have to perform an RNA extraction, and that uses up reagents for RNA extraction, it uses up time, and it uses up resources, so it reduces the turnaround time, increases the turnaround time of the assay. And so our workflow is actually extraction free. It's pretty neat. It means that you can go really directly from the saliva without performing that RNA extraction. Eliminates a whole other reagent bottleneck that takes place, and it means that you can go directly from the saliva. And I'm just checking the box for any other questions, and it seems that we've covered everything that's came through today. So with that, I think we'll stop there. Thank you again for your time. And my colleague Anita, I think, is going to now close out the webinar. So thank you so much for attending. We appreciate it.
Thank you, everyone. Thank you, Mark. We will follow up with everybody with a follow-up email. Please look forward to that. We will include the PDF of today's presentation, as well as a link for on-demand viewing. Have a good day, everyone, and good evening.